and Mike were like my parents. So I still remember I was standing here November 99, was standing here. I remember the last words I said is, when I left, I probably won't be protected and probably won't be protected by this roof. I still remember the words. So I spent probably half of my time in the library. So I was sitting in the library for a while. And I realized, I told Mike, I said, this is probably the 6th year, the best time of my life. Different from what you perspective, but I think this life-wise, I think it's my best life. And I really appreciate the help you gave to me, and all the other colleagues and staff, the support they gave to me, and to make me as today. I really thank you very much. This time there was an exchange. It's an exchange between the Chinese Academy of Engineering and U.S. Academy of Engineering. It's a symposium. And then I realized this is your one, so I really want to get into that. So there's a couple of rounds of selection. So I finally get to be a participant. I finally a speaker. Only both U.S. side and the Chinese side, there are only 2 speakers. So I really feel an honor to be here. So mostly today's talk is similar to the talk I gave yesterday. I gave the last talk over the symposium. Although I would like to tell them more about how use your one, but I think I probably just right now focus on the science part, and we're going to leave the rest of the warm talks at the private talking.
Do you have a laser pointer?
Yeah, I think I need a laser pointer. Where it is? Yes.
All of you that sticks.
It was the tradition, yeah. I know. Yeah, where's the stick?
Yeah.
It's over there. Yeah, what's the? It's the same stick? Wow, same stick. Yeah. So my presentation basically divided into 2 parts. The first part is the in vivo counting. That's the first part, and then the second part is the in vivo trapping. So this is actually the extension part of my PhD thesis here. I was doing trapping with Tatiana at that time. And then I'll tell you why I get into that part. So first, the in vivo counting. So I think it should be this part, yeah, yes. So the motivation to get us to do in vivo counting is when we study cancer metastasis. Cancer metastasis is really a complicated processing. Thank you. It's typically if you have there's a primary tumor, then after a while the tumor cell is not satisfied with local environment and get into a circulation. And by the theory of seed and soil theory, and the cancer cells, circulating tumor cells, will stop at a particular place, and after a while gets in the distant site and become a secondary tumor, which we call distant metastasis. So that's the whole step. And currently thinking is 80% cancer patients die of metastases. So spreading is a really key issue. And secondly, probably the survival is really depends on the early diagnosis. For the circulating tumor cells, the clinicians think probably that's the only one so far that's a measurable and early indication of the cancer metastasis. So that's why we are interested in measuring this, quantify the circulating tumor cells. The current way to quantify the circulating tumor cells is usually it's by in vitro method. For example, typically it's a flow cytometry, it's in vitro flow cytometry. And there's also a supplementary technique that's a confocal two-photon can do a supplementary tool, and also the regular fluorescence microscopy. But there are limitations. First of all, in vitro method, we cannot do dynamic monitoring, especially for animals. Typically for a mouse, there's a 2 milliliter blood in the whole body of mouse. You cannot draw blood all the time, otherwise the mouse will die. And it's hard to do real-time quantitative measurement because the in vitro not only has to do blood, you have to separate and do the purification, labeling. All those takes time. It typically take a couple hours, and sometimes it could take days. It got low sensitivity because not all the cancer cell has certain marker. Currently, the typical marker is epithelium marker, but not cancer cell have that biomarker. I'm not mentioning it also depends on the blood drawing. So what we're trying to do is we're trying to find a non-invasive method to monitor and quantify the circulating tumor cells. Firstly, in animals, and later probably in the human being, which I'm going to tell you today. Now, optical molecular imaging has been advancing very fast for the last 10 years. Although this modality allows you to see vessels, and sometimes can see the detail of vessels, but so far it cannot provide analysis information in terms of the blood component. For example, what kind of cells in there, what kind of biomarkers in there. So far, optical molecular imaging cannot do that. This was a video, but since it was copied as it's not a computer, so the video is gone, but it should be fine. The idea of this video is, the left one is, we can usually see the blood, the vasculature over the mouse ear. It's a live mouse, of course. So this is an artery, this is a vein. And this is like the daily choke of our lab. So we can see this easily. And the right side is supposed to be a video. That's fine, because the video shows, this is a real-time video, we can see some of the cells is flowing. So the video is actually showing when we label the tumor cells in vitro, for example, leukemic cells in vitro, and we injected those cells intravenously into the mouse. And when we look at those mouse ear, this vasculature, you will see those labeled tumor cells actually flowing. And flowing both in this is vein, and also the artery. But typically they go anti-directional. And we can see the cells actually flowing here. You can see one cell's here, one cell's here. But can we quantify those cells? can we quantify those flowing tumor cells? It's hard. It turns out it's very hard to quantify those circulating tumor cells by just imaging method. Even for this real time, which means 30 frames per second, even with that fast, we can see the cells, but the problem is the blood flow is very fast. The first frame, the cells might be here, and the second frame, you might out of the frame. So it's hard to just analyze how many cells pass by a certain place. Therefore, it's hard to quantify the circulating tumor cells, mainly because it just flows so fast. So when our optical people have the responsibility to solve this issue. So then this reminds me, and this is work when I was done in the Harvard Medical School environment lab. So both Charles Lee and I were thinking, how are we solving this problem? So we thought about to design an in vivo flow cytometer. So the idea is this. And by the way, I think the reason I do in vivo flow cytometry is both cytometry and imaging are the two frequently used instrumentation when I was a graduate here. So that's why I think I ended up doing this. So that's the reason. So this reminds us the in vitro flow cytometer, which is right outside the biomedics lab. So we have if we put a weak laser spot over a mouse ear, this is artery, and we make it relatively rectangular shape from typically a round shape Gaussian beam. And then this becomes a slit. Then if a labeled fluorescent cells pass by that slit, then will give a burst. It can be collected by the objective, the microscope objective. Then we have a signal. So this is more like a in vivo version of the flow cytometry. A flow cytometry is just a flow shoes, artificial flow shoes, and then the cells flow in the artificial flow shoes. So this is the original data we are looking at. This is 30 seconds of the fluorescent signal out from that slit collected by the objective. And the y-axis is the intensity of the fluorescence out from that slit. So you can see those peaks. Those peaks represent wherever a fluorescence labeled cell pass by that slit, then it will give a fluorescent burst. Mainly due to the different labeling of the cells, the differences give different high intensity of the peaks when they pass by that slit. So the task here become we just count those peaks out of the noise. Then we have a count, say how many cells pass by that slit per minute. So that's become this kind of question, measurement. And typically, one peak has two parameters. One is intensity, of course, it's got fluorescence. And secondly, it has this width or duration of the time. So this is seconds. So typically, a positive peak has 5 to 10 milliseconds. And it's corresponding to the time duration the cell pass through that slit. The slit has the dimension approximately, this dimension is 70 micron, and then this direction is the flow direction. It's this width of the slit is approximately 5 micron. So that duration of time corresponds to the time passed by the slit, so which is approximately 5 to 10 milliseconds. So this is enough to separate those signals from either sonar noise or electronic noise. So we published those results 2005 in Nature. We can monitor those circulating leukemic cells. When we inject those fluorescent labeled leukemic cells, we see those cells deplete from the circulation and start early metastasis, and we can see this depletion curve. This is the cells in the circulation. And 1 out of 50 reagents we tried, one agent called SDF1 delayed that depletion. And this delayed depletion may be related to the prevention of cancer metastasis, because the cells stay longer in the circulation, and actually much less cells gets into the tissue. So this become a method can do a quick, non-invasive screen of what kind of drug can affect the circulation and prevent this metastasis or inhibit the metastasis. So this could be a tool to do such screening. And this is what look like on the optical table. This is right now we have a much nicer version. This is the early time of my setup in Fudan University, and these have become much smaller by now. So it more or less look like a microscope. So this we call it in vivo flow cytometry. It actually more look like it's a microscope. And this is inverted microscope. You can see an objective here, and the mouse is on top of it. And so this in vivo flow cytometry. We call it in vivo flow cytometer. Is basically it's a hybrid of imaging and the in vitro flow cytometer. And the optical setup look like this. So first we use CCD, we put the mouse on, we see where the vessel is, and we turn on the laser. And laser it goes through a cylindrical lens to make a slit. Also has 2 confocal slits to make sure we're looking only that vessel. We are only looking one single vessel all the time. This could be over a month. But this slit is to narrow down that Z direction, make sure we are looking on only one vessel, not the vessels above and below that particular vessel. So we only look in the one vessel. And then the laser excitation over here, and the mouse is here, and when it's after the excitation, then the fluorescence coming back, go back to the PMT and get collected. So this is in the experiments. It look like this. This is the anesthetized mouse, and underneath the mouse is the objective, and this is the mouse ear, and this is the vasculature, and we choose relatively a big vessel close to the ear, the body, because we want to still see a bigger vessel. Bigger vessel means bigger volume of blood, means we can have a high chance to measuring those circulating tumor cells. And this is typically we're looking at artery, because artery first it's flowing relatively fast, and secondly the flow is relatively constant compared to the vein. After we got the novel instrumentation out, we still have some challenge to make the biologist to use the setup, mainly because the analysis, it still takes time, and sometimes it's hard to do. And the biggest problem is require user interaction, means because we rely on principal component analysis, the intensity and the duration. So this is a typical PCA analysis look like. Usually, we used to just, before any real experiment, we have to do a control. A control mouse, a control data means without injecting any reagents or cells, you just inject blank serum. And this is your background is. So this usually take an hour. Just get the control, negative control. And then you start your experiments. The signal look like this. Then you draw a line, show how many positive cells in contrast to negative control. But sometimes it's hard to draw this line. It's like this, or like that, or like this. It's hard. It's similar to a problem encountered by an in vitro flow cytometer, which is the gating. And not mentioning, it's also require user interaction because we have drawn a line, so which takes a lot of time. Music is nice. And so we're trying to tackle that origin problem, because if we want any instrumentation to be accepted by a biologist, we need to make it simple and easy to use. So then we are trying to develop the algorithm and software. First, it's automatic, and secondly, it's user-friendly. So after trying a couple of algorithms, we found out the wavelet-based denoising method was pretty good in terms of reducing the noise and for cell picking. The reason for wavelet-based denoising, I think, my personal feeling is, thanks for the biological phenomenon, which is pretty much consistent. Because for the first 30 minutes and next 30 minutes, for the biological phenomenon is pretty much occurring in a predictable phase. So that's why the two part of the signal look like more or less similar. That's why wavelet-based denoising method is pretty good at this kind of case. And then this is what the results look like. So this is the golden standard cell picking. So just by visually, we can see this is a cell, this is a cell, this is a cell, and then we count 83 peaks. And this is the cell picked out by the automatic wavelet denoising method. And you can see the cells has been picked out by this wavelet-based algorithm. And you can see pretty good agreement between the golden standard picking method and the wavelet-based denoising method. But the outcome of this is almost real time. So basically, when the user finish the acquisition, 1 or 2 seconds, the user will get a number. Say this 83 peaks for the first. This is probably 3 minutes something. So instantly the user will get a number without doing anything. So that's much friendly to a user. And also this is automatic. So the user likes it. And then this we developed when I came back to China 87, 07, 08. And then we also gave the code to Charles Lin. And Charles Labs also use that automatic counting method. It's much easier and much quicker. And then the next step we're trying to do is to make sure this works better for the biologist. especially for the cancer study. Although this in vivo flow cytometry can use not only in the area of circulating tumor cells, it can be used in all kinds of circulating tumor cells as long as it's labeled. But so far, we feel the circulating tumor cells is probably one of the best applications because circulating tumor becomes such an important study object. And one of the complaints from the biologists, from the cancer people said, the mouse model we used before is an injection model, which means we have to label the cells in vitro and then label them and they inject into the mouse ear, into mouse by the intravenous injection. And the cancer people said, this is not the best way to do a cancer study. The best way at this moment is the orthotopic tumor model. So they challenged me and said, can you do that? Can your instrumentation do that? So I said, luckily you have both background from the optics and the biology. So we built a tumor model. And in collaboration with a medical doctor, we built this orthotopic tumor model. This is liver tumor model. And when we build a model, typically we injected 500 cells to start with. When we inject the cells has green fluorescent protein DNA into it. So when they grow, they have GFP, green fluorescent protein, so they are fluorescent. So when they grow, they have fluorescent. When they start to metastasize, the true circulating tumor cells also has fluorescence, so which solves the problem of labeling. So we don't have to label outside, like by the in vitro labeling of the cell culture method. So this is nice. So, and then this is more or less mimic the pathological condition, so which is adopted by the cancer field. So we built this model, and we monitor the circulating tumor cells. Now we have fluorescence. We have green fluorescent protein into it. So after 2 to 3 weeks, we start to see the rise of number of circulating tumor cells, and the rise exponentially this week. So this is to us was very exciting. This is probably the first time. This is the first time we received this rise, and this correlated well with the growth of the tumor, the size of the tumor. Previously, people thought that's the case. The bigger the tumor, the more circulating tumor cells it should have. But this is the first accurate measurement of the numbers, and it was top published cancer research. And then we did an application to show. Now we have a method to monitor the circulating tumor cells in vivo continuously, means monitoring the cancer metastasis, at least early indication of cancer metastasis. And we did an application. One of the challenge to medical doctors is the resection. With that cancer resection will cause metastasis. So some patients, if they do surgery, if they don't do tumor surgery, they probably can survive 2 years. At least a fraction of patients, when they undergo the surgery, they start to have problem of metastasis, and then they probably have survival of only 7 months. This is in the practical sense. So the question becomes, will that surgery cause metastasis? So we did this animal model. We show this is without the resection. With the resection, the number of circulating tumor cells keep low, at least for 1 month or 2. And to simply translate this animal study to human study, it's a very naive, simple translation. It's times 12. So if this happens in 1 or 2 months in mouse, this could be, the results can be applied in human is 1 year to 2 years. So this, if we do this liver removal, surgery removal carefully, then we'll be fine for 1 year or 2. We don't have cancer metastasis. After this study was published in Cancer Research, some medical doctors come to me and said, it's not fair because the liver, you can remove the tumor completely. But some tumor is hard to remove completely, do a complete surgery. For example, pancreatic cancer. Because the shape is weird, it's hard to remove all the suspected tumor site. So if this is the case, if some tumor site was left by the surgery, would that cause more metastasis at all? So we are trying to answer that at this moment.
Is the cover with resection you have CTS?
With the resection, this keeps low. Without the resection, it just keep growing. Yeah. Maybe I was get reversed.
Sorry, I think it's information. Was recession done at 4 weeks?
No, this is the resection just down by 1 day. This is just let the mouse grow by the 4 weeks, and then we do the surgery. The 4 weeks is just for the growth, yeah. And then to make this technology more usable possibly for clinical use, we also built a photoacoustic version. Photoacoustic have the advantage of label-free for some cases, and also can have deeper detection. So at least for now, we found the right application for circulating tumor cell detection, which is melanoma. Melanoma has melanin. Melanoma cell has melanin. Melanin has strong photoacoustic signal upon certain wavelength excitation of laser. So when the cell in the circulation, they undergo a laser excitation. They get the absorbing light, the cell absorbing that specific light, wavelength of light, and then get it heated for a few tens of degrees, expanded, and then give out acoustic signal. So that's a principle of this photoacoustic detection. And so far we found out for the tumor case, the melanoma has this strong photoacoustic signal. And in the mouse model, we successfully measured the number of circulating tumor cells by the photoacoustic flow cytometry. And this is the animal, implant the tumor, and then we got a measurement of the number of circulating melanoma cells along the week. And this time, it does not have labeling at all. It's only rely on the endogenous signal from the melanin. So this could be, if it's not killing, but the first medical application of circulating tumor cell counting in vivo and continuous monitoring, and possibly to evaluate the response to therapy. And just switch the gear a little bit, just for I don't want to take too much time, probably for the next 10 minutes or so. I want to take on the second part of my talk, which is immunotrapping, which means manipulating the cells by optical traps in the live animal. When I was a graduate student here, we basically play this kind of games all the times for fun. And these are cells, and those optical trapping, I don't want to spend too much time on explaining. So this institute is very, very unique to in this planet. If we use the trapping to trap the cells, if we move the stage, other cell move with the stage, and then because this cell was trapped by the optical trapping, so this becomes a stationary. So if we move the laser, the cell will go with the laser and then become a manipulation tool as well. And about 2008, 2007, I was thinking, 10 years, almost 10 years after I left here, I thought, and then I spent 10 more years on optical, in vivo counting. At one time I thought, if we can do in vivo counting, can we do in vivo trapping? If we can count them, why don't we just trap them out of the circulation in a live animal? So that's become the motivation at that time. And previous, all the studies are in vitro. Oops, since this is another computer, so the first video cannot play out. I hope the second video will work. So basically, the challenge for the in vivo trapping is, this is mouse ear. So the skin is highly scattered tissue. But to maintain a trapping force, a terminology called gradient force, maintain the Gaussian beam of a laser is very important. So laser is a Gaussian beam. To maintain the shape of the Gaussian beam is very important. But the skin is highly scattered tissue. So if the laser goes into self-focused into the skin, it will be distorted. So it means the Gaussian beam will be also distorted, and it reduces the trapping force. Reduced into a significant sense, then you won't have a trapping inside of the animal anymore. So that's a challenge from translating from in vitro to in vivo. And after 2 years, we're trying to maintain the shape of the Gaussian beam and the focus beam, and finally we can trap them in vivo. And this is what it shows. So firstly, this class is where the trapping is, and this is the mouse ear. This is the capillary. It's the mouse ear. So when we turn on the optical trapping, first we see a slowdown of the blood flow, and then we slow down to a case, then we can see some individual cells. See, and then finally we can trap the cell. We can trap the cell. And then the second video, I think it's going to work out. And then not only we can trap one cell, we can also trap a group of cells, and which will block the flow of the capillary. So this is trapping a few cells. And you can see this cell is going to the trap, and those trapping of those couple of cells finally block the blood flow. So as optical people, we have to, if we block the capillary, this Chinese saying is, you have to solve the problem, to unblock it. So that's the old Chinese saying. So then we have to unblock it. So next we're gonna unblock the flow. So this is blocking, and we use a trapping. We try to trap the front cells and get them one by one, get them out one by one, and we try to restore the blood flow. This is clearly look like it's RBC. You can see the shape. And we're trying to use the claws, the trappers, to take the first cell away out of the trapping force. And this is real time, by the way. And so we trap the first cell, we try to get it away. And finally, we take the first away, and the second away, and then restore the flow. I'm going to just stop a minute. Now you can have a question. So I think that's the last slides, and this is the person I want to thank for the collaboration, and this is the grants I got. It's part of the grants I got from China. Thank you very much for your attention.
Thanks, Xunbin.
Yeah, can you go into exactly how you conducted the in vivo trapping? Like, you have to maintain your Gaussian beam as you go towards the...
Yeah, this is what our exactly trapping is. The typical depths of, this is 3D trapping, by the way. The typical trapping depth is approximately 40 to 60 micron in the skin. But we think it could be go deeper if it's a relatively transparent tissue. There are a couple of ways to maintain the Gaussian shape. You have to tap it. That's why we tried 2 years. Firstly, we just cannot do it. And secondly, we also currently trying to waveguide to make it a better maintenance of the Gaussian shape. Right now we're trying that, and then we're trying to have a deeper trapping of the cells. Yeah.
Can you selectively trap the red blood cells, or you trap other cells?
That's the next step we're going to do. At least right now we cannot do specific trapping. We think we might be. There are a couple of ways to do it. For the tumor, if we have a relatively larger fraction, we might be able to trap them, because the tumor cells are definitely going to have different shape. But it's challenging, because in the blood, larger portions are red blood cells. But we're trying that. We're trying to do a specific. That's our next step. Yeah, Jim.
So what do you see as the applications for trapping?
For the research-wise, I think manipulation, the stem cell, might be a good application, because there are other types of trapping as well, in addition to optic. There are magnetic trapping, also used in animal studies. There are also AFM, but AFM cannot be used in vivo. It's hard. Magnetic traps are used in animal study, but cannot choose cells and cannot choose locations that elegantly. So for this type of application, I think because stem cell to manipulating real cells is the advantage of this type of technology, and which I think is the stem cell. So right now, to have stem cell implantation at specific location is very important, at least for understanding how it works and where to start to grow is very important. So I think the stem cell might be a very good application of this. Another is to study microcirculation. For example, study how the atherosclerosis starts. After this paper was published, I was invited to give a talk in the International Society of Microcirculation, out of my surprise. But they are very interested, because similar to study as atherosclerosis, people want to know when the cells stick to the wall and can you remove it. And current model is to use drug to mimicking this process. But the problem of use drug to induce such a model is once you have this drug, you have this model, it's already late. You can only monitor late stage. You cannot see the early process of these pathological processes. And this way is a very elegant way to induce such pathological condition, and you can watch the processes, and also you can reverse it. So I think that's a possible application. But for clinically, some doctor told me they're excited. They said, this can be used for surgery, which I become very conservative because for medical application, I think that this is a little bit far away because the force-wise and complication of the medical process, the procedures, I think this probably is far away. Mike?
Can you discuss the labeling procedures used for in vivo flow cytometry? I'm wondering whether it's possible to do surface antibody labeling or something like that. Is there sensitivity there?
What can you say?
Surface
Self?
antibody.
What is self-antibodiable?
If you label particular cell types with antibodies,
Okay.
then you might be able to make things more applicable to human.
Right. The labeling is it's a big part of this kind of research. So right now we have different labeling methods. The early part is for the injection of the tumor cells, as I already showed.
Labeling we're using for that.
Yeah, and for labeling for example, lipid and for injection. And second labeling is to do this orthotopic model, the genetic labeling. And third,
So we don't have GFP tagged foods.
right, thirdly, we could use antibody. We can inject fluorescence antibody into the circulation. We can label them approximately after 5 to 10 minutes. We have those data. I didn't show it because this is not a full story, so I didn't put up those data. But it could be. You just injected fluorescence labeled antibody. We can see the cells. But one complication depends on the full antibody or FC portion. These cells can be depleted. So, but we could, it's not a problem. And once we actually tried is this published where we monitor the circulating apoptotic cells. So we use Nexin-5. This was, I think, Ralph Westletter. So he has a tag, use Site5, Site7, Nexin. Then we can see the number of circulating tumor cells, the apoptotic cells decreasing from circulation, and very fast, out of a half time of 6 minutes. So labeling is not a problem, and plus the circulation actually is a good way to mix for mixing. Our prediction, my personal feeling is approximately 5 to 10 minutes, the mixing going to be very good. So that's not a problem. Actually, one colleague, my Chinese colleague, told me, because he has a very good antibody for circulating tumor cells, so he would like to try that in vivo. One exciting result recently we have is we found much larger fraction of circulating tumor clusters in the circulation. Circulating tumors have become a hot topic for the last 10 years, and clusters become a hot topic last year. I published a Cell Nature Science paper, a couple of them, and showing the cluster might be very important. And by the in vivo study, we recently got the data. We show the cluster might be 3 times fold than we thought, than currently people thought. People thought the most probably 9%. But at some time point of the tumor growth, the fraction of the cluster become dramatically increased. So although our very strict criteria, we measure about 30%, especially at the middle phase of the metastasis. This which might be helping the metastasis. And we are submitting that to, I think, clinical oncology something. But I think it's possible. Very exciting.
Xunbin, just a question about the tweezers.
Yeah.
So, based on your knowledge of the force generated and the typical forces in a trap, does it make sense that you're actually trapping in a 3D geometry in a true potential well, or are you taking advantage of radiation force pressure in a tube, in a cylinder? So are you actually pressing the erythrocytes against that cylinder and causing some fractionation of the flow partial stoppage of the flow because of the radiation force pressure.
And that could be interesting. That could be an interesting application. So far, we haven't thought about that. What we demonstrate in this paper is that the trapping for the in vivo case is also three-dimensional. So, for example, if we focusing up and down of the vascular, we can see the other fields become blurry because we're defocusing. But the cells go with the focusing, so it means the trapping is also three-dimensional. For that one to combine with the radiation force and also the wall, and to have a combination of different processes, mechanical processes, yeah, that would be interesting. We haven't thought about that. We haven't tried it, but we probably can talk about it a little bit later. Yeah.
And for the in vivo flow cytometry, are you moving towards any clinical applications, and are there better targets for looking at vasculature, for example, in the eye?
Yes. So there are two directions we're trying to do. One is we're trying to look at the eye. And secondly, we're trying to not only monitor melanoma cells in vivo, but also can expand that to a general types of tumors. So eye might be a way to go. And thirdly, we're trying to also couple that to therapy. So if we can count them, we can trap them, can we also treating them? Use either mechanical or thermal or some combination with the photodynamics therapy. Yes, that's the direction we want to go. Yeah.
Thank you. So any more questions for Xunbin? Okay, let's thank him again.
Thank you.
These are on the rest of the day, so please let us know if you'd like to
Thank you.
talk. Thank you. It's a good talk.
Yeah, I think one time probably we should do more collaborations. Because I think, well, a lot of common interest. So it's still